Doratha Byrd, and she is actually a fresh faculty in the Moffitt Cancer Center, just joined in January of 2021. And she's currently an assistant member in the Department of Cancer Epidemiology, Gastrointestinal Oncology in the Moffitt Cancer Center. So Dr. Byrd received her BS in biology and then MPH in epidemiology from the University of Florida. After that, she went to get her PhD in epidemiology in Emory University, where her dissertation research really focused on the development and validation of a novel inflammation biomarker panel, weighted dietary and lifestyle inflammation scores, and their association with the colorectal neoplasms. So I think, again, we have really, the colorectal cancer is probably the best one studied in terms of the impact of microbiome. So that's great. Now, in January 2019, she joined the NCI Division of Cancer Epidemiology and Genetics as a postdoctoral fellow. And during her time there, she conducted methodologic microbiota studies and investigated association of microbiota with cancer risk and diet with the gut metabolome. In Jan 2021, she joined Moffitt Cancer Center as an assistant member in the Department of Cancer Epidemiology, and where she will continue to contribute to the reduction of cancer disparities using the integrative interdisciplinary approach to study microbiota-mediated mechanisms for cancer risk and among diverse populations. I think she's already having some discussions with the people who joined this hub meeting, which is great, and I think really begins the first day of her collaboration with others, because the first time she will be discussing her work and probably will connect with the others in the hub, and that will be a good collaborative environment. With much ado, I'd like to invite, I owe the floor to you, Dr. Byrd. Please go ahead.
Thank you. Thank you so much for having me. And I'm really excited to be here today and looking forward to the possibility of many collaborations in the future. So today I just want to talk about how I would like to kind of set the stage for innovative studies, microbiome studies to address cancer disparities in the future, hopefully using an integrative and interdisciplinary approach. So I always like to start off with a bit of background about why I do the research I do and why I am here today. I then want to briefly cover methodology for studying the microbiome, which I think is so important if we really want to address the associations of the microbiome with any disease endpoint. I'll then go into the associations of the microbiome with disparities with colorectal cancer and with breast cancer, focusing on a few examples in the literature in my prior work. I then want to speak briefly about microbiome-related metabolites and how I think they can somewhat address the limitations of the existing data and biospecimens collected in the microbiome field. I then will talk briefly about some future directions. So as background, I was born in Dothan, Alabama. So I spent a lot of time with my grandmothers pictured here. So they're now 82 and 90 years old. But my grandfathers, they actually died quite young. One, in fact, from cardiovascular disease at the age of 39. And what kind of drives me is that, I think a lot about the collective trauma that my grandparents went through while they grew up in the South, where, especially at the time, the systems of oppression and discrimination were very heinous. But one thing that has always stuck with me about my grandmothers was how they kind of found something that they could take control of in their life to improve their quality of life. And for them, that was their diet. So as I got into college and my master's program, and I became very, very interested in diet as well, and I was learning about the vast health disparities that exist in the United States, I began to think about how we can use diet as a potential mechanism to improve health among populations that have been disproportionately affected. So over the course of my master's and PhD program, there was a lot of research coming out characterizing the structure and function of the microbiome, implicating the microbiome in cancer risk and etiology. And also, there was emerging evidence that the microbiome could be modified through diet and lifestyle exposures. So my research program today focuses on this connection through the microbiome of the associations of diet with cancer. And I'm hoping that I can one day be able to make broader implications toward cancer disparities with this etiological research. So what is the gut microbiome? So I'm sure most of you are fully aware, but this is, these are the trillions of bacteria and other microbes that inhabit our GI tract. They have many different physiological functions, especially in one of the areas of my interest is for their role in cancer. And I think what makes the gut microbiome an exciting target for cancer prevention is that these bugs are potentially modifiable, such as through diet and lifestyle exposures. Now, I briefly want to touch on methodology for how we can study the microbiome. Usually in order to study the gut microbiome, we collect either tissue or fecal samples. Colon tissue may have some advantages in that it may characterize the bacteria that have invaded the colon crypts. And we're conducting some tissue studies here at Moffitt using currently available samples. We can also use fecal samples. And I want to pause for a moment and acknowledge that how we collect these samples is extremely important. And this is because we know that the collection method used may impact what we observe downstream in terms of our microbiome metrics. Now, ideally, we want a sample collection method that preserves that original microbiome signature and that stabilizes the microbiome DNA, such as during when participants are shipping their samples into the lab or the samples are stored at room temperature over the course of multiple days. I also think that microbiome researchers should start thinking about developing a common collection method, or at least having one common collection method in common with other microbiome researchers. And I think that if this were to occur in the future, this would really facilitate data pooling. And in order to address diversity, we would have address disparities, we would have a more diverse data source if we're able to pool microbiome data. So some of the work that I did at the National Cancer Institute focused on methodology for collecting fecal samples, and I'll just give a brief example here. So we collected fecal samples from 15 volunteers at NCI. And then we aliquoted the sample from each individual into 4 different collection methods. And we aliquoted within those collection methods one sample that was frozen immediately and another sample that was frozen after being left out at room temperature for 4 days. We then metagenomically sequenced those samples. And what we found was that 95% ethanol didn't do such a great job. I did maybe a moderate job of stabilizing the microbiome, but FIT tubes, FOBT cards, and RNA later did a great job of stabilizing the microbial DNA over the course of 4 days. Now, FIT tubes and these FOBT cards are colorectal cancer screening tools, and I'll get into that in future slides. But I think that these colorectal cancer screening tools may open up opportunities to establish diverse prospective microbiome cohorts using or leveraging existing colorectal cancer screening programs. So after we've collected our microbiome samples, they undergo sequencing. Broadly, there's 2 different types of sequencing, 16S sequencing, which doesn't provide too much detail. We only get taxonomy down to the genus level, but this is great for hypothesis generating studies, and generally it is much cheaper than shotgun metagenomic sequencing, which sequences all of the DNA in the sample, and we can assign the taxonomy all the way down to the species level. We can ascertain genes and functional pathways, but it is more expensive. And I think this is really where the field is headed. So after we've sequenced our DNA, usually a bioinformatician will take those sequences and conduct some complex algorithms to cluster those sequences into representative units that we can then link to taxonomy, and we can use them to create various diversity metrics. So the two metrics that I'll primarily focus on are alpha and beta diversity. So I'll briefly go through these. I know many of you are familiar with these metrics, but so alpha diversity is our within sample diversity. So thinking about how many shapes or how many bacteria are present in a sample and what is the distribution of those bacteria. So for example, for our middle circle here, we have 4 different shapes and they're evenly distributed. So we can say we have a richness of 4 and a high evenness. Whereas if we look at the first circle, we have 2 shapes and there's an uneven distribution. In my studies, we typically focus on observed sequence variants to characterize richness, the Shannon index to characterize both richness and evenness, and face phylogenetic diversity to incorporate evolutionary history. Generally, what you'll see in the literature is that higher alpha diversity is associated with lower disease risk, but I want to acknowledge that I think there's a lot of work to be done in this area still, and that may not always hold up to be true. Beta diversity is our between sample diversity. So if we want to compare person A to person B and compare the shapes between the two, we see that there are, for example, 2 shapes that are in person B that are not in person A. So we can say that the beta diversity here is 2. There are many different metrics, again, to characterize beta diversity. The ones that I tend to focus on is Bray Curtis distance, which just characterizes differences in microbial abundances between 2 samples, and unifrac-based measures, which can incorporate some measures of differences in evolutionary history between 2 samples. So when we want to calculate beta diversity, usually we want to do so between each pair of samples. So we will put together a distance matrix for each pairwise comparison. And then we can do many things with this distance matrix. One of the things being that we can sort of project the matrix into like a multi-dimensional space and plot principal coordinates, such as what was done here. And we can color those coordinates to look for some clustering by certain characteristics. Switching gears now, I want to talk about why the microbiome may be a great tool in my mind to really address disparities. So we know that race is a social construct that was really created, rooted in justification for oppression and discrimination based on how someone looks. We also know that there's issues with ethnicity as well, because we tend to group very heterogeneous groups of people into one ethnic group. But we consistently see that there are certain exposures that are associated with someone's self-reported race or ethnicity, such as racism and the downstream psychosocial effects. There may be differences in diet and lifestyle and other environmental factors. The issue is that many of these exposures are very complex and oftentimes very difficult to measure. But these exposures are likely acting individually or maybe interacting to affect gut microbiome composition. And we know that the gut microbiome is in turn associated with many of the most disparate conditions that are leading contributors to mortality in the United States. So I think that this makes the gut microbiome a great target to be able to understand potential biological mechanisms underlying disparities. And there has been some literature demonstrating differences across racial and ethnic groups in the microbiome. For example, there was a cross-sectional study conducted in the Southern Community Cohort where they characterized the oral microbiome among African Americans and European Americans. They found differences in alpha diversity, differences in beta diversity, and also differences in abundance of certain taxa. But I do want to note a prominent limitation of this study is that they were not really able to conduct a detailed investigation into certain exposures that might have been driving these racial and ethnic differences in the microbiome. For example, they didn't have detailed information on periodontal disease. And we know that periodontal disease is more prevalent among African Americans. Periodontal disease is associated with many disparate conditions. And we know that periodontal disease is strongly associated with the oral microbiome. So I think if we really want to conduct meaningful research comparing the microbiome across racial and ethnic groups, we really need to dig into these exposures that might be driving these microbiome differences and potentially driving downstream disease. There is another study conducted in Amsterdam, Netherlands, where they compared the fecal microbiome across 5 different ethnic groups. Again, they found differences in alpha diversity, they found differences in beta diversity. For example, you can see some clustering here. And they found differences in certain taxa. And in this study, they tried to do a great job of teasing out these ethnicity-related factors that could be driving these associations, but the differences still remained. So I think in the future, it's important to continue to characterize the fecal and oral microbiome across diverse racial and ethnic groups and geographic locations and other socio-demographic factors. So as I mentioned earlier, the FIT test is a colorectal cancer screening test that we previously found at NCI to be a stable and accurate way to measure the microbiome. And I am hoping that we can characterize the microbiome in these FIT tests in diverse populations to be able to accomplish that goal of characterizing the microbiome in diverse populations. So though I'm hoping to create a FIT biobank, which would be a prospective cohort of these FIT test residuals collected from diverse participants in the clinical screening populations here in Florida and nationally. Now to do this, what I will need to do is leverage existing clinical colorectal cancer screening programs. I'm currently working with the Tampa Bay Community Cancer Network and investigators in the health behaviors branch here at Moffitt to work with community clinics to collect these samples. And if any of you have any other ideas for great places to collect colorectal cancer screening samples, I would certainly love to hear from you. So what the study design would kind of look like is that the patients who are normally scheduled to have their FIT test would go to their appointment as normal, and they would be given their FIT test as normal, but they would also be provided some questionnaires where we can ascertain important factors like antibiotic use or major recent medical conditions. A subset will be piloted with a diet and lifestyle behavior questionnaire, and we will also collect detailed medical history data as well. So then the subjects will take their FIT test home. They will collect their stool sample as normal and complete the questionnaires. The FIT test will be brought back to the clinic for hemoglobin or blood testing as it normally would. The real difference here is that after that FIT test is tested for blood, we would have coordinators go to the clinics to collect the residual test to be brought back to Moffitt for aliquoting and freezing. Now, the kind of nice thing about FIT testing in terms of characterizing the microbiome is that anyone who tests negative on their initial FIT test is recommended to repeat testing every year. So we would potentially have many repeat samples collected prospectively over the course of multiple years. And the ultimate goal would be to link these individuals to various disease endpoints, such as to clinic records, cancer registries, and the National Death Index. So I think this would create a very rich resource for those interested in microbiome and disease research. So thinking in terms of short and long-term impact, I think that there's going to be hopefully so much that can be done with this data, even just in terms of characterizing the microbiome in this somewhat unique population, perhaps developing risk prediction models and cross-sectional case control studies. And there's many long-term values of this study as well. It'd be great to characterize the trajectories in the microbiome over time and to be able to investigate the longitudinal associations of the microbiome with cancer risk, which would be a major improvement on the existing, mostly cross-sectional data in the microbiome field. So switching gears now, I want to talk a bit about my work and prior work on the microbiome in breast cancer. Colorectal cancer. So there is strong plausibility for the associations of the gut microbiome with colorectal cancer risk. We know that many established colorectal cancer risk factors, such as obesity and certain dietary factors, likely influence the composition and function of the gut microbiome. And in turn, the gut microbiome has many different etiological effects that likely increase or decrease risk for colorectal cancer. And colorectal cancer is a particularly prominent cancer in the US, as it is the second overall leading cause of cancer death. And we also know that there are extensive disparities existing in colorectal cancer. So, for example, we know that colorectal cancer incidence is about 20% higher among Black Americans compared to white Americans. And colorectal cancer mortality is about 40% higher among Black Americans compared to white Americans. And though we can see that the overall trends of colorectal cancer incidence and mortality are decreasing, the trends among individuals younger than the age of 50 or early onset colorectal cancers are actually increasing. And the disparities, the racial and ethnic disparities are still very present in these early onset cancers in terms of likelihood of survival. And we can even think of the recent tragic passing of the actor Chadwick Boseman. Now, I think that the gut microbiome, again, is an exciting target to study disparities in colorectal cancer. Again, there may be many exposures that are associated with race and ethnicity that influence the composition of the microbiome, in turn increasing or decreasing risk for cancer. So I haven't gotten to conduct too much work on the gut microbiome and colorectal cancer disparities quite yet, but I do want to highlight a study that I really liked that I would hope to expand upon here at Moffitt. This was a case control study of colorectal cancer where they measured the colon tissue microbiome in African American and non-Hispanic white colorectal cancer cases and controls. And they used a lot of targeted sequencing methods to target specifically sulfur-producing bacteria. I'll just focus on one today, Bilophila wadsworthia, which is a hydrogen sulfide-producing bacteria. And one thing that they found was that among African Americans collectively, compared to non-Hispanic whites collectively, there was a much higher abundance of this bacteria. And when we looked at when they investigated the associations by race, they found that this bacteria was strongly positively associated with odds of colorectal cancer among African Americans, but the associations among non-Hispanic whites were weak to null. So, I think that it is, again, plausible that microbiome and colorectal cancer associations could differ across race and ethnicity, but I think we need to really work on replicating these findings. And so here at Moffitt, what I'm hoping to do is use existing tissue specimens to compare the microbiome and colorectal cancer associations across racial and ethnic groups. And also, I'm very interested in comparing the microbiome across early onset versus later onset colorectal cancers. Switching gears again, I want to talk a bit about the associations of the microbiome with breast cancer in my prior work in this area. Again, breast cancer is a very disparate cancer. So even though Black women are less likely to develop breast cancer, they have a higher risk for mortality from breast cancer. Also, Black women are more likely to get certain cancers that are more aggressive in nature, but even among the more treatable subtypes of breast cancer, mortality tends to be higher among Black women. And I think that the gut microbiome could be a great target for potentially understanding disparities in breast cancer. There's strong plausibility for the associations of the gut microbiome with breast cancer risk. We know that the gut microbiome may have roles in hormonal regulation and also regulation of immune and metabolic functions, all of which could increase or decrease risk for breast cancer. There's a lot of gaps in the literature when it comes to the associations of the gut microbiome with breast cancer. There's really been few previous studies, most with small sample sizes, and there's been few studies that really had the detailed histological and immunohistochemical characterization that is needed to really dig into the associations of the gut microbiome with breast cancer. There's also been no studies conducted in Africa. And I think that this is a very important population to conduct this work in because we know that breast cancer incidence and mortality is starting to increase in certain parts of Africa, and the studies focus specifically in sub-Saharan Africa. We also know that African ancestry women are more likely to be diagnosed at younger ages, at later stages, and with more aggressive subtypes of breast cancer. So to fill in some gaps in knowledge in the field, we conducted a study, we leveraged data and biospecimens from the Ghana Breast Health Study. So this was a population-based case control study. And in this study, the cases presented to 3 hospitals in 2 large cities in Ghana. They had their suspicious breast lumps biopsied, and the controls were selected from the areas that gave rise to the cases, and they were frequency matched to the cases by district of residence and by age. Now, many of the cases and many of the controls provided stool samples for which we used 16S RNA sequencing to characterize the fecal bacteria. I present here the associations of alpha diversity with breast cancer. To orient you, we divided participants into tertiles based on the distribution among controls and used logistic regression to estimate odds ratios. And overall, we found that there was a strong inverse association of alpha diversity with breast cancer. For example, there was about a 66% lower odds of breast cancer comparing those in the highest relative to lowest tertile of the Shannon index. And then we also found that these associations were very similar by hormone receptor status and also by cancer grade. And we also found strikingly similar associations for non-malignant breast diseases as well. Now, to provide some context to these findings, alpha diversity, we know, may have many roles in estrogen deconjugation. So we could hypothesize that alpha diversity may contribute to overall estrogen burden. And we know that estrogen is mechanistically linked to hormone receptor positive breast cancers. And we also could hypothesize that alpha diversity has many roles in immune function and metabolism, as demonstrated by considerable literature. So we present here some beta diversity associations. I just focus here specifically on Bray Curtis distance. So this is just a principal coordinate plot of the first two axes of the of the Bray Curtis distance matrix. And we can see here some clustering by case data. So the controls tend to cluster separately from the breast cancer and non-malignant breast disease cases. And we conducted multivariable test called Meerkat to assess the confounder adjusted beta diversity associations, finding that these beta diversity associations were statistically significant. So I present here the associations of relative abundance of selected taxa with breast cancer. So relative abundance is just the number of reads of a taxa in one sample divided by the sum of all reads in that sample. And we found, for example, that there were multiple taxa that were either positively or inversely associated with breast cancer, one of them being bacteroides. And for every 1% increase in relative abundance of bacteroides, there was about a 5% higher odds of breast cancer. So to provide some context, we know that many of the taxa that we found that were associated with breast cancer may encode enzymes that are involved in estrogen deconjugation, therefore potentially contributing to overall estrogen burden. But this was a study that only used 16S sequencing. We really need shotgun metagenomic studies to dig into the functionality and to ascertain the microbial genes that were present in these samples. Some of the taxa have also previously been shown to be involved in bile acid and short-chain fatty acid metabolism. So in conclusion, it's plausible that the gut microbiome may be strongly associated with breast cancer. But again, I'm emphasizing a replication in large, diverse populations here in the US. In the future, we really need studies that focus on functionality, such as ascertained through shotgun metagenomics. We need detailed immunohistochemical, histological, clinical characteristic data, some of which was lacking in the Ghana Breast Health Study. And we also need prospective studies to rule out reverse causality. And this could be done potentially in prospective cohorts using existing blood samples measured by measuring microbiome-related metabolites, which I will talk a bit about now. So a limitation of many of the existing fecal microbiome studies is that most of them are cross-sectional, and they also tend to lack diversity. So most of the studies have been maybe case control studies where they measured the microbiome and compared it between people who have cancer and who do not have cancer. But the issue is that you can't really tell whether the bacteria are driving the cancer or is it the cancer driving the microbiome. We haven't really been able to really tease out those associations. So ideally, what we would want is to have a prospective cohort, like, for example, what I hope to develop with the FIT Biobank, where we collect multiple fecal samples sequentially over time and follow participants prospectively until they develop cancer. But we don't really have that now. Now what we really have is one blood sample typically collected at baseline and maybe one point sometime later on in most of the existing prospective cohorts here in the United States. So using these blood samples, we perhaps can measure metabolites that are related and potentially produced by the microbiome to better understand how the microbiome may be involved with cancer risk. So some of the microbiome-related metabolites of interest for me are TMAO and its precursors, choline, betaine, and L-carnitine. Now, choline, betaine, and L-carnitine can be derived from diet or they can be endogenously derived, and they all contain these trimethylamine substrates that upon reaching the gut microbiota can be cleaved to form trimethylamine, which travels to the liver to form TMAO. Now, you may have heard of TMAO mostly in relation to risk for cardiovascular disease. There's some very convincing evidence in that space, but there's emerging evidence that choline and TMAO and perhaps some of the other trimethylamine containing compounds may be associated with risk for colorectal cancer. So what we're doing to kind of address, again, some of the limitations existing in the microbiome field using these microbiome-related metabolites is conducting nested case control studies where we measure TMAO and its precursors among individuals who did and did not develop colorectal cancer over time. So this will allow us to conduct a prospective analysis, and I think we'll also be able to really add to the literature by conducting a meta-analysis of the associations of TMAO and its precursors among African Americans. So those assays are currently underway. We're waiting for the results. I wish I could present them to you today, but in the meantime, to better understand these associations, we're taking a dry data approach by investigating the associations of dietary choline and betaine with mortality in the NIH AARP cohort. And we plan to do a similar analysis for colorectal cancer, but we're waiting for some updated colorectal cancer incidence data. So I'll just present this to you today as a preview for hopefully what's to come in the future. So the NIH AARP cohort is a large prospective cohort of men and women. And at baseline, all of the men and women completed a diet history questionnaire from which we were able to calculate dietary choline and betaine. We took those values and divided them into quintiles. So here I present a sex-specific analysis. And so, for example, comparing those in the highest relative to lowest quintile of total choline, we found about a 6% higher risk for all-cause mortality, a 12% higher risk for all cardiovascular disease mortality, very consistent with the previous literature, and about a 6% higher risk for all cancer mortality. Among women, the associations were generally much weaker and close to the null. For dietary betaine, which has in the literature been demonstrated to mostly have some protective features, we again calculated dietary betaine at baseline and divided it into quintiles. And we found about a 3% lower risk comparing those in the highest relative to lowest quintile of total betaine. This would be for all-cause mortality. About a 7% lower risk for all cardiovascular disease mortality, and no association really for all cancer mortality. Among women, we found slightly stronger inverse associations for all-cause and all cardiovascular disease mortality. We found about a 7% lower risk for all-cause mortality among women and about an 11% lower risk for all cardiovascular disease mortality. So in the future, we are planning to calculate choline and betaine in other large cohorts, and we will conduct a meta-analysis of these findings. But we really are going to need to combine this data with microbiome and other targeted metabolomic data, I think, to really be able to make some progress towards getting toward some better answers about the associations of choline and betaine and TMAO with mortality and cancer risk in the future. Another metabolite of interest is bile acids. So primary bile acids are derived from cholesterol, and they can be conjugated to glycine or taurine in the liver. Now, bile acids that escape enterohepatic circulation and reach the gut microbiota can be transformed to form secondary bile acids. And secondary bile acids have long been hypothesized to have pro-carcinogenic and disease-promoting effects that could potentially increase risk for multiple different cancers and other diseases. Now, similar to TMAO and its precursors, there's likely a role of diet in the synthesis and excretion of bile acids. Fat has been shown to promote synthesis of bile acids, whereas fiber has been shown to promote excretion of bile acids. And there's strong in vitro and animal evidence for this, but the evidence for the associations of diet with bile acids among humans has been really limited. So to further investigate this, kind of taking a cancer prevention approach, we used cross-sectional data from a subset of 2 large cohorts. One was a cohort of Finnish male smokers, and one was a cohort of United States men and women, and they both collected blood samples. So I present here the associations of just soluble fiber with bile acids to orient you. We divided participants into quartiles of fiber based on the distribution among the controls. And this data was from a nested case control study. And we used linear regression to estimate proportional differences in bile acid concentrations across those quartiles. And for example, we found that there was about a 74% lower concentration of taro-ursodeoxycholic acid, comparing those in the highest relative to lowest quartile of soluble fiber. And I presented here taro-ursodeoxycholic acid and glycocholic acid specifically because these are also bile acids that were previously associated with colorectal cancer risk. So as I mentioned, these were 2 separate populations that we studied, and they were quite different in terms of their population characteristics. So for the rest of the components that we investigated, alcohol, coffee, and fat, there were some kind of inconsistencies in the associations that we observed. We found that in ATBC, or among those Finnish male smokers, alcohol, trans fat, and polyunsaturated fat was positively associated with bile acids, but coffee and monounsaturated fat was inversely associated with bile acids, but again, only in those Finnish male smokers. The corresponding associations were generally weak or null in the PLCO study, or among those United States men and women. So as you may have noticed here, there was a difference in fasting status of the blood collected. And I just want to note this because I think that it is plausible that there could be true population differences. As I mentioned, the gut microbiome is involved in generation of or the gut microbiome is associated with bile acid regulation and synthesis, and we know that the gut microbiome could plausibly differ across various geographic locations. So there could be population differences, but we really weren't able to tease them out due to these methodological differences of, and how the blood was collected. So I just want to emphasize here how important it is to have consistent sample collection so that we in the future can be better able to tease out population specific versus methodological differences in our studies. So as a future direction, we're currently analyzing bile acid concentration data in a study called the Polyp Prevention Trial. So this was a randomized intervention study of the effects of a high fiber and high fruit and vegetable, low fat diet. And just as a preview, we are finding that there are a few associations of a few effects of the intervention on certain bile acids that were, again, previously associated with colorectal cancer risk. And we plan to conduct similar studies with TMAO and its precursors. And I want to take the time to, again, emphasize the importance of diversity. So, in order to really get to this goal of identifying these targetable exposures that could be driving disparities, we really need to have a diverse study population to be able to do so. So in the future, what we really need are large population-based studies that are prospective in nature in diverse populations with microbiome samples. And I'll emphasize diversity again here because we know that this has been a huge problem just broadly in science. So for example, in 2016, we know that 81% of the genome-wide association study data was generated from European ancestry individuals. Only 3% was generated from individuals of African ancestry and 0.05% from indigenous populations. So I'm hoping that in the microbiome field, we can do a better job of improving diversity in our study populations. And then to do this, we can't do it on our own. It's really going to take a multidisciplinary approach to be able to recruit diverse participants into our microbiome studies. We need to work with community stakeholders, with behavioral scientists and clinicians and other individuals to really understand how to best recruit diverse participants into our studies and develop targeted and appropriate study materials and communication procedures with these participants. And we also need to do a better job of understanding the potential barriers that participants may have in participating in our microbiome studies. So ultimately, I think to really make a change in reducing disparities, I hope that with more diverse populations, we'll be able to really meticulously identify these targetable exposures that are driving disparities, and we can really develop interventions to be able to reduce these exposures and really make a change. I have many people to acknowledge, and it's been great at Moffitt Cancer Center. I've met so many wonderful mentors and collaborators, and I hope to meet many more at USF. And thank you so much for having me. And if you don't want to say anything today, I'm happy to take additional questions and connect via email or Twitter.
Thank you very much, Dr. Byrd, for this interesting and very exciting presentation. I think we have time for some questions, and hopefully, you will stay on just to answer them. Some people might get in touch with you by email or even chat. But let me start with one question. I know that there is a large data set with the AARP, NIH AARP, with a diet, but we know that the colorectal cancer also coming into the younger age group, early onset, between 30 and 50. And is there a data set for that as well, so that one could actually look at, what are those factors that actually begin the process in terms of going into colorectal cancer later on in life?
So that's a great question. So that's been, one answer I've been hoping to find myself, and I haven't been quite successful in terms of existing data sets. But here at Moffitt, there are a lot of early onset colorectal cancer cases. So I'm hoping that we can begin to, collect samples from these, collect stool samples from these participants and use the existing data that we have to at least kind of address some of these questions. It's going to be difficult to address questions about risk but I'm hoping that these data sets will be coming along in the future, and if anyone knows of any existing data, I'd be happy to learn more about them as well.
Yeah, thank you. Any other questions from the audience? You could click your hand button, or you could, that would be a good way. Yes, go ahead. Joseph, go ahead.
Hi, Armen. Thank you for your presentation. With the stool collection methods, I was wondering, did you compare to, for the frozen sample, did you homogenize the frozen sample to compare? Because there's For instance, papers showing you can see a difference in the microbiome just on whether you are homogenizing your sample or not, since different parts of the stool would favor different microbes, such as anaerobes being more in the center, aerobes more on the surface.
Yes. So for our freshly frozen samples, and for our 95% ethanol and in RNA later samples, we did homogenize. It's a little trickier, I think, with the FOBT cards, but we did do our best to homogenize for those as well. And I'll have to maybe talk.
So how if you're collecting on a card, how can you homogenize?
Yes, so I have to talk to, I would probably have to defer that to the lab a little bit, because I know we did a lot of validation work to figure out how best to work with the cards, because there are some different considerations that you need to have. So I think that is something that I can follow up with you with, or if you want to send me an email, I can.
Okay, yes, I'd like to know. Thank you.
Dr. Yu? Ji-Yoon Yu. Yeah, go ahead.
Hi, it was wonderful presentation. I have one question. You measured the TMAO and choline and betaine related to cardiovascular disease and then colorectal cancer mortality. Have you ever chanced to looking at TMA producing bacteria compositions or other bacteria composition between those groups? Because I think the TMAO is converted by bacteria. So.
Yes, and so that's actually one thing I'm hoping to do in the future. I haven't had the chance to do that yet, but I think that maybe the associations of dietary, choline, and betaine with cancer, with colorectal cancer, might be modified by these TMA producing bacteria, as you mentioned. So that is something that I'm hoping to be able to do as a part of a future grant.
Thank you.